Hi, hello, namaste everyone. So today in this video, we are going to know about the procedure to find out the different factors which are required for photosynthesis or not. That means in the absence of that factor, whether photosynthesis occurs or not. To prove that, we have to perform some experiments. So in this video, we are going to discuss the procedure of that experiment. Okay, so let us start with one experiment and the procedure how to perform. So first one, before going to know that, can you tell me the overall equation of photosynthesis? Okay, so tell me 6CO2 plus 12 H2O gives rise C6H12O6 plus 6H2O plus O6O2. Okay, so here we will write light and chlorophyll. Light and chlorophyll. So these are the reactants and these are the end products. Okay, so here in the presence of, that means green plants, means which possess chlorophyll, in the presence of light, by utilizing carbon dioxide and water, they synthesize glucose, and along with that water as well as oxygen also generated. Okay, so this is the overall equation for photosynthesis. If these four are available, then plant can synthesize this glucose and then that's stored in the form of starch. Okay, when these four factors are available, then only this will form. Now let us take first factor, carbon dioxide. So in this experiment we are going to prove that, means by performing this experiment, we can prove that without carbon dioxide plants cannot synthesize food. Okay, so here how to perform this, the procedure involves in this, now we are going to discuss. So first of all, we have to take one potted plant. So we need a plant growing in a pot and which possess a long narrow leaves. The plant which possess long narrow leaves. So such kind of plant we should select for this experiment, which possess long narrow leaves. So this kind of plant we should select for this experiment. Then first step in this experiment, what we have to do is, we have to keep this potted plant in the dark room for 3 to 4 days. 3 to 4 days we have to keep this plant in the dark room. Why? Because to remove the starch which is present in the leaves. How it is removed? How means here new food material is not inside, the remaining which is present in the leaves, that is utilized by the plant, so that we can make the leaves free from starch by keeping the plant for 3 to 4 days in the dark room. Okay, then after 3 to 4 days we have to take this plant out. Here we need some other apparatus to this. So what is required? We need a bottle. We need a transparent glass bottle. Transparent glass bottle is required. Okay, so like this, a transparent glass bottle is required for us, and along with that a splitted cork is required to keep here. Okay, splitted cork, so that we can insert through that slit or this leaf into the bottle. Okay, so here first of all we have to take this bottle, transparent glass bottle, and we have to take some potassium hydroxide into the glass bottle. Potassium hydroxide solution we have to take into this bottle, 5 to 10 ml of potassium hydroxide. Why? Because to remove the carbon dioxide which is present in the bottle. Okay, so we have to take 10 ml of potassium or 5 ml of potassium hydroxide into the bottle. Then we have to take this bottle and through this split, okay, we have to insert one healthy leaf. We have to insert healthy leaf into the glass bottle through the split cork. Through the split cork, we have to insert like this. With the help of stand, so we have to hold this glass bottle. Okay, so what we do here, KOH, potassium hydroxide solution we have taken here to remove the carbon dioxide which is present in the bottle. Okay, we have to insert only half of the leaf into the bottle. Okay, half leaf should be outside, half leaf should be inside the bottle. Then at the rim of this, we have to apply Vaseline to prevent the further entry of air into the bottle. Okay, so like this we have to arrange all the apparatus. So what we did here, for 3 to 4 days we kept this plant in the dark room to remove the starch which is present in that leaves. Okay, so after that we took a glass bottle which possesses a splitted cork, and we took KOH solution into that to remove the carbon dioxide. Okay, then we have to insert like this half of the leaf into the bottle through that splitted cork. Okay, then this whole apparatus we have to keep under the sunlight. Under the sunlight we have to keep for 3 to 4 hours. Okay, so we have to pour water. We need to pour water to this plant. Then we have to keep under the sunlight. Okay, now what we have to do after 3 to 4 hours? After 3 to 4 hours, we have to remove this bottle and we have to take this leaf. We have to detach the leaf from the plant. We have to take that detached leaf. Outside from the bottle, and we have to detach the leaf from the plant. Then we have to immerse this leaf, detached leaf, into the boiling alcohol. So into the boiling alcohol, we have to immerse this leaf, and so that what happens, all the pigments which are present in that dissolves. Chlorophyll pigments, mainly chlorophyll pigments, dissolves in the alcohol, so that it turns to pale color. Leaf turns to pale color. Okay, then we have to take out the leaf and we have to wash with water. Then we have to add few drops of iodine solution on the leaf. Okay, when we spread the iodine solution on the leaf, so here we can find the half of the leaf which is present outside the bottle. This portion, it turns to dark blue color. You know, the iodine test used to test the carbohydrates. No, when there are carbohydrates, that turns to blue color. So that's why here in this region the leaf synthesized carbohydrates. That's why it turns to dark blue color. But which is present inside the bottle, that won't turns to blue. Why? Because see here. So this part of the leaf which is present outside the bottle, so it receives light and also it receives water, chlorophyll is present in that, and from the atmosphere it receives carbon dioxide. Everything, all the four it receives, which is present outside the bottle, right, which is present inside the bottle. So since it is transparent, so it receives the light, so light is available to that, and chlorophyll also present, and water through the xylem vessels it reaches. But carbon dioxide is absent. Why? Because we removed carbon dioxide by adding potassium hydroxide solution into the bottle. Since carbon dioxide is not available to this part of the leaf, so this portion of the leaf did not synthesize food. Understood? So in that way we can prove that, so without carbon dioxide plants cannot synthesize food. Okay, so this portion received everything. That's why this portion turned to dark blue, because here in this region food is synthesized, but not here, because carbon dioxide is not available to this region of the leaf. Okay, so from this experiment we can prove that without carbon dioxide photosynthesis cannot take place. That means plants cannot synthesize food. Okay.